Next speaker is Dr. Iain Haslam from the University of Manchester, United Kingdom. He analyzed the animal model. The title is SHA signaling regulates the damage response of murine and human hair follicle in chemotherapy-induced alopecia.
Okay, thank you very much. No disclosure. So I'm just going to talk about a study that we've been looking at, the role of sonic hedgehog in the pathogenesis of chemotherapy-induced alopecia. Now, chemotherapy-induced alopecia is still a significant problem for chemotherapy patients, and it's a particularly traumatic side effect that faces a significant psychological burden on the patients. And just to briefly refresh everyone's memory about chemotherapy-induced alopecia, the chemotherapy generally targets the rapidly proliferating cells of matrix keratinocytes. This pushes the follicle down one of two pathways. If the damage is small, the follicles will go down the dystrophic anagen pathway. This will result in hair shedding and a primary recovery of the hair follicle in which a new hair shaft, poor quality hair shaft, and often depigmented hair shaft is produced. If the damage is severe, then the hair follicle will be pushed down the dystrophic catagen pathway, in which hair loss is generally worse, but the primary recovery is more rapid. Now, there are still no real effective treatments for chemotherapy-induced alopecia beyond scalp cooling, which does work for a subset of patients. However, the lead academic in this study recently showed that in the chicken feather, sonic hedgehog signaling is critically involved in chemotherapy-induced feather damage. And what he was able to show is that if you add cyclophosphamide to chickens, you get this damage in the feather barbs. And this produces a memory, effectively, in feathers, which shows when damage occurred. He was able to show that sonic hedgehog signaling was critically involved in this. And the damage occurred in these barbs where sonic hedgehog expression was high, but not in the central rachis where sonic hedgehog expression was absent. So moving on from this study, we sought to determine whether these same mechanisms exist in mice and man. So using a non-biased next-generation sequencing approach, we looked at the expression of gene expression following 1 and 2 days post-cyclophosphamide treatment in anagen-induced mouse skin. And importantly, the sonic hedgehog pathway was identified to be the most disrupted pathway with downregulation of this signaling. So to determine whether it was the sonic hedgehog signaling pathway itself that was resulting in the hair follicle damage, we looked at using a sonic hedgehog signaling inhibitor, cyclopamine, which binds to smooth and prevents downstream signaling in the sonic hedgehog pathway. And what we found was that, strikingly, inhibiting sonic hedgehog alone produced the sort of effects in the hair follicle that you would see following chemotherapy-induced damage. So proliferation was reduced, apoptosis was increased, and you saw a hair follicle phenotype that looked remarkably like a dystrophic catagen follicle, with hair follicle dystrophy, follicular distortion, and melanin dystrophy. So now that we'd found that actually just inhibiting sonic hedgehog alone could create some of the damage that we see following chemotherapy-induced alopecia, we wanted to see whether we could rescue this effect with the addition of exogenous sonic hedgehog. So after depilation to induce anagen, mice were treated with either cyclophosphamide or cyclophosphamide plus a recombinant sonic hedgehog. And I'll draw your attention to 11 days post cyclophosphamide here, and these higher powered panels. And what you can see is cyclophosphamide treatment on its own resulted in the hair loss that you would expect from chemotherapy. However, the addition of recombinant sonic hedgehog actually rescued hair growth, but strikingly, it didn't rescue any pigmentary defect, which suggests that the loss of pigment is decoupled from any sonic hedgehog signaling. Obviously, we wanted to understand whether this pathway was also applicable to human hair follicles, and we used the well-established Philpott method for culturing human hair follicles ex vivo. And looking at 4-hydroxycyclophosphamide, the active metabolite of cyclophosphamide, doxorubicin, another drug commonly known to cause chemotherapy-induced alopecia, and the sonic hedgehog pathway inhibitor, we could see that you get downregulation of sonic hedgehog pathway genes looking at qPCR. We also did a very small pilot study looking at plucked hair follicles from patients undergoing various chemotherapy regimens. And I'll draw your attention to this column here, which shows that downregulation of sonic hedgehog gene expression in these plucked hair follicles was a striking correlation to actual hair loss suffered by these patients. This is a very small pilot study, but does provide some interesting insight in vivo data. Looking again at the ex vivo human hair follicle organ culture technique, we wanted to see if we could replicate the results that we've seen in the mice using this model. And indeed, if you look at cyclopamine inhibition of the sonic hedgehog signaling pathway, you indeed see a reduction in proliferation, an increase in apoptosis, and melanin clumping around the hair bulb, and this is in the absence of any chemotherapy. Again, looking at the potential rescue of the hair follicle damage, we treated these follicles with 4-hydroxycyclophosphamide in the presence and absence of recombinant sonic hedgehog. And we were able to show some slight rescue of the phenotype. So proliferation was still reduced when you had the recombinant sonic hedgehog in there, but not to the same degree as when 4HC was on its own. There was a significant decrease in apoptosis with the addition of the recombinant hedgehog, although we didn't see any change in total melanin levels, which again agrees with the mouse data, suggesting that any melanin damage is decoupled from sonic hedgehog. So to conclude, what we've shown is that, as with chicken feather, sonic hedgehog signaling seems to be intimately involved in the damage that we see following chemotherapy in the hair follicle. And the next step will obviously now be to identify clinically applicable sonic hedgehog agonists that can be evaluated in vivo to potentially protect against damaging effects of cyclophosphamide and other chemotherapy agents. And moving on from this, it's possible that sonic hedgehog may be involved in a number of other side effects from chemotherapy, notably GI syndrome, memory loss, hemocystitis, and infertility. So I'd just like to make some acknowledgements. Thank you to Ralph House for involving me in this project. Zitao Yu was the lead academic, and these are our other co-authors in this study. And of course, thank you to Nilüfer Investment Parjo for access to human hair follicles, without which we wouldn't be able to do most of our work, and funding from Manchester. Thank you.
We have about 4 minutes for questions. Anyone want to stand up at microphone 1, 2, or 3 and ask any of the 5 for any questions now? We will have the last 4 after this, and we will start again after we've asked the question. Anyone standing up and screaming?
Erin, do you have any experience compare the capsule and the DP cell in the assay? Do you have experience compare the capsule and DP cell in the other half story?
Yes, we didn't do any in vivo studies with cup and papilla cells. We've done quite a lot with in vitro mixed lymphocyte assays, and the results are very similar. So the cup cells and the papilla cells both seem to be very immune privileged. Sheath cells, non-bulbodermal sheath cells, are not nearly so effective. So they may have some activity, but not as much.
I'd ask Dr. Tobin or Kevin if they're any closer to finding the exact target of alopecia areata site of damage.
That's a very good question. Initially, I was convinced about the melanocyte, but it took a while to move away from melanocyte. I think there's a differentiation relationship with alopecia areata. We have a targeting at a particular stage in early anagen, So there's something happening around the building up of the follicle that is releasing or making more accessible to the immune system the target antigen. But I think it's a normal protein that may be biotransformed in a particular way at a particular time, and with the susceptibility of the individual, it perhaps pushes them over a threshold. I think it's a thresholding thing that may explain why part of the scalp may have active loss, part of the scalp may have active regrowth, and part of the scalp may have no change. I think there's a thresholding effect in terms of how the immune system is recognizing a differentiation associated with antigen.
Do you think it, maybe as Dr. Prowse proposed, a phenotype and not a genotype? Response?
I've no idea.
Can I follow that question? Angela, sorry. Des, that was beautiful, but I missed it because I didn't know we were changing the program.
Sorry.
We've been spending a lot of time looking at parallels between rheumatoid arthritis and alopecia.
Can't hear you, sorry.
We've been looking at similarities between rheumatoid arthritis and alopecia areata. So there are trichohyalin autoantibodies in rheumatoid arthritis, but to my knowledge, trichohyalin is not expressed in the synovium. So I just wonder what you think about that when you look at it from a clinician point of view. And also, it ties in with the question of these huge size polymorphisms in trichohyalin, the gene. There are tremendous chunks of amino acids that differ in the population. So, I don't know. I'm struck by this discovery in rheumatoid arthritis, and yet it hasn't really been addressed. Do you think this is just antibodies that all of us have?
I think your first question is very appropriate, because I think it's not actually trichohyalin itself. It's a motif on trichohyalin that's shared across a whole raft of different targets. That changes also with citrinylation, when you move arginines to citrulline, and we see across all of these different conditions very high levels of arginines. In trichohyalin, it's super high, at about 27%, and I think this involvement of the PADs, the peptidyl arginine deaminases, and then also the transglutaminases, is changing the conformations of these proteins, making them more sometimes soluble and potentially more easily presented in the MHC context. So I don't think the focus is on trichohyalin. I think the focus is on a motif, or an epitope on trichohyalin that is shared by nature of the amino acids that are there. And the second one, sorry.
Yeah, that was good. How about your comment about pigment cells? Have you moved away from thinking of melanocytes as a target?
I think the pigmentation side was more because of the most vulnerable stage in early anagen to attack the hair follicle. And we are very clear that the reconstruction of the pigmentary unit is very subtle in the early stages of anagen, say 2, 3, and 4. And we know that melanin itself, if it's not particularly well organized, can be particularly reactive. And but Andrew Messenger and others have shown many examples of people who don't have selective targeting of pigmented hair as being a feature of alopecia areata. So it's not an exclusive link. I think there may be some link, and maybe a differentiation associated link within a stage of anagen, maybe 3, 4. But also trichohyalin is a very early differentiation marker too, and a rather exposed one in the early stages.
Can I make one more comment?
Can we move on? We're about to run out. Number one.
A question for the last speaker who talked about the chemotherapy-induced alopecia. Have you looked at other chemotherapeutic agents, and particularly Taxol, which would be microtubule mediated?
No, we haven't at the moment actually. So we've been using cyclophosphamide primarily because that's the model that's been most commonly used, particularly in the model that Ralph Pass developed in the 90s. So at the moment, we're restricted to just one agent.
Yes, because I would suggest that might be a very different mechanism.
Well, that's very true. I mean, obviously most chemotherapy classes have different mechanisms of damage. So we believe that they could all eventually signal through from the catch-up, but that needs to be determined.